Hello, my name is Vincent. Today we'll be discussing cancer, its origin. In this presentation, I'll be given a high-level view of the origins of cancer. In particular, I will be discussing the main aberrations, such as gene mutations, specifically gene mutations involving oncogenes and tumor suppressor genes. Also, I'll be discussing the defects of DNA repair, not in much detail, but just an overview of that one. And then talking about immortality, avoiding apoptosis, and all these lead to cancer. At the end of the discussion, I will also be briefly talking about angiogenesis and metastasis. So we begin here. Why? So why does cancer occur? This is a question that will be answered by the end of this presentation. I'll be discussing the main mechanisms, the main themes, the main processes that lead to cancer. So let's begin with the cell, an individual cell. And this is a normal cell. So each normal cell can become cancer. There's no master controller that determines what all the cells will do. There's no hierarchical system that controls all the cells in the body. In a sense, in a healthy body, the cells carry out a well-orchestrated autonomy and a harmony. They are beautiful craftsmen in which each cell builds its designated part without an overarching foreman. And although each cell can become a cancer cell, for the most part, cells that become cancers, or the majority of cells that become cancers, are those that are divided, that divide most often. And those would be cells found in the lungs, the skin, the bone marrow, sex organs such as ovaries, uterus, breast, prostate, testes, cells in the bladder, cells in the kidneys, and the gastrointestinal tract. And those make up the vast majority of cells of the cancers. That constitutes roughly 90% of the cancers in those areas that I just mentioned. So where do these cells get their information? How do they know what to build? So that begins, the cells know what to build and which tools to use and when to use them because of a blueprint, a detailed instruction manual. In this case, the instruction manual is within chromosomes. There's 22 pairs of autosomes or chromosomes, so 1 through 22 as shown here. They're numbered by size, so the first pair is bigger than the second pair, the third, and all the way down to the 22nd. So you can see the 22nd is much smaller than the first. But these are pairs of chromosomes. The first 22 pairs are considered autosomes, which means both men and women have them. The last one that you see here is XY. Those are sex chromosomes. XY means that it's male, and XX would mean female. So in this case, this is a male set of chromosomes. So there's 22 pairs that we all have. There's 23 total, of which the 23rd is a sex chromosome. So if you multiply the 23 by 2, you end up with 46 individual chromosomes. Each chromosome, one you receive from your mother, one you receive from your father. This is DNA, a picture of DNA or a replica of DNA. So why do I show you DNA? So what's the purpose of the DNA? So the chromosomes that I showed you in the previous slide, the chromosomes are made up of DNA. And within this DNA, the double helical structure, are the instructions that I was alluding to in the previous slide. The instructions basically come in little packets of information called genes. And these genes are the instructions of how, when, and what a cell should do. These instructions tell the cell how to grow, when to grow, what to grow to, how many times it should be replicated, when to stop if there's a problem during the replication process, how to repair the process, and if the cell is damaged or the DNA is damaged too much, how to commit suicide in a process called apoptosis if there's too much damage. It also tells normal cells, in a sense, normal cells express only those genes that maintain the cell type function or maintain its particular cell type. Cancer genes, in contrast, express genes or don't express genes or basically become cells that they're not supposed to become. Ultimately, genes are instruction manuals. That's the instruction manual. And ultimately, those genes express or become proteins, or they give the instructions of how to build proteins. In this slide, I basically give an overview of what we just talked about. So we start out with the chromosomes. Remember, there's a 22 that we all have, and then the extra one with either X or Y or XX with male or female. So we all have chromosomes. The chromosomes are made up of DNA, and then within the DNA is the gene. And so we're going to be talking a lot about genes. Chromosomes, as I mentioned, I should mention this. There's estimated 20,000 genes. So that little pink part that's shown here, there's estimated 20,000 genes in the human chromosomes, all those chromosomes that I showed. Prior to completing the Human Genome Project, there was thought to be 100,000, but that number is obviously reduced, and there's some speculation that it may even be reduced smaller than 20,000, but there's 20,000 genes. So, and again, the genes produce proteins. If we go a little bit, magnify the DNA a little bit closer, we'll find that the DNA is made up of a sugar phosphate backbone, and that's the blue ribbon there in this helical structure. But within that ribbon, there are what are considered base pairs. And the base pairs are a specific orientation. There's only 4 different base pairs. If we go back to our analogy, there's only 4 letters used in these instructions. And the letters are A, T, G, C. A standing for adenine, T standing for thymine, G standing for guanine, C standing for cytosine. These base pairs are in a specific arrangement. The A is always paired with T, so adenine is always with thymine. And so the guanine is always paired with cytosine. And that's universal in all the DNA. So what's the purpose of these base pairs? What do they do? Well, they're basically, the instruction manual. They're the words, if you will, of what is to be produced, which is ultimately proteins. So let's look at this a little bit more in detail. So on top, you'll see the gene again. Within the double-stranded DNA, there's the gene. And the gene, if you look at it, magnify it, you're going to find that what's considered the sense strand of DNA. The sense strand of DNA, we'll see those GTs, GTCs, A's. And of those, they're put into a specific arrangement that gives the instructions for what protein to make. And they're put in what's called a triplet. So one strand of DNA, again, is made of the sense protein. And from that sense protein, you make an RNA, or in this case, it's specifically an mRNA sequence. The sense strand is read in groups of 3, like I said, groups of 3 called triplets. Each triplet produces a codon in the mRNA. The pairing of the DNA and RNA remains the same as a DNA, is that you'll see a G with a C, an A with a T, and a G with a C, but then the next one you see an A and a U. So on RNA, the U replaces the T. So normally in DNA, you'll have ATs and GC combinations. But in RNA, instead of a T, you'll have a U. So that's why on that 4th grouping, the start of the second triplet, the A on the sense DNA, if you look right below that, you'll see the mRNA has a U. But then the G's and C's always match up, whether DNA and RNA. It's just the A and the U in the DNA. The U is only found in the mRNA. So what's the purpose of this codon? And the purpose of that codon is to tell what protein, what amino acid to produce. So we'll go to our next slide and get more specific. So each codon tells which amino acid to produce or to hook on. So in this case, we have on the codon, we have GUG. So if you go first letter G on the table there, and then you go the second letter, which says on the top second letter, you go to U. So we're in the first column, very bottom. And then you go to the third letter, which you can say the third letter up on the top, and then you go all the way down, and you go to the G. So GUG is a valine. So that's a particular amino acid. Proteins are made of a string of amino acids. We go to the next one, which is CUG. You start with C on the first letter, which is to the far left, and then you go to U, which again, second letter up at the top, far left again, and then we go to the right-hand side and see third letter. Third letter says it's a G, and the G, so a CUG is a leucine. Again, another amino acid. If we turn to a whole string of mRNA, we'll see valine, histidine, leucine, threonine, proline, glutamic acid, glutamic acid. That's a string of amino acids. When you put thousands of amino acids together, what you end up getting is a protein. And that's ultimately what we're looking for. And so why does it matter? I mean, why do we care what the DNA looks like? Okay, the instructions that we're giving, the genes that we're talking about, they give instructions to produce a protein. So what? What does that matter to us when it comes to cancer? So it matters because we want to know what's happening to the DNA sequence in cancer. And that's the key point. The key point is what are we going to find out? And what we find out is that in cancer, there's a mutation. A mutation is a change in the DNA. So if there's a change in the DNA, that sequence that we're talking about, the DNA itself, that sequence of DNA changes. When that sequence of DNA changes, the mRNA changes. If the mRNA changes, then the protein can change. And if the protein changes, what we're going to find out is that that's what potentially can lead to cancer, depending on what the protein changes into. Because those proteins oftentimes are associated with growth or stopping growth, either starting growth, continuing growth, stopping growth, or some element, in cancer in particular, some element of the growth process. So mutations, again, are changes within the DNA. They can be detrimental if it occurs within a gene that controls a specific aspect of the growth. And these are called oncogenes. There's a mutation that can also happen in one of the inhibitory genes, which are the tumor suppressor genes. There's also what we'll talk about in a while here is in the DNA, so that DNA replication and repair process, when DNA is replicated, when basically made a copy of it, there can also be some mutations in the repair. So again, mutations are when there's a change in the DNA. So the question may be then, well, why do mutations occur? I mean, why don't they occur more often? Why do they occur now? Or why do they occur in particular situations to particular individuals? So we'll start with the easiest one. So there's two types of agents, or two definitions that we need to know. One's called mutagens and one is called carcinogens. So what are mutagens? Mutagens cause mutations. So what we were just talking about is that you get changes in the DNA. If there's a particular thing that causes changes in the DNA, those are called mutagens. If they're carcinogens, which just means a particular element or agent can cause cancer. So mutagens cause mutations, which are changes in DNA. Carcinogens can cause cancer. All mutagens are carcinogens, but not all carcinogens are mutagens. Let me say that again. All mutagens are carcinogens, which means all agents that mutate DNA are carcinogens, but not all carcinogens are mutagens. So there's agents out there, and we'll get to them. Example being alcohol, which doesn't change the DNA, but it can still cause cancer. Again, carcinogens cause cancer. So we'll start with an easy example. So chemicals, radiation are mutagens. Particular chemicals, not all chemicals, particular chemicals are mutagens. Radiation is a mutagen. So by definition, some chemicals and radiation cause changes to the DNA. Mutations, back to our analogy, mutagens change the instruction manual, change the gene. The changes in the instruction manual, the gene, this is called a mutation. And radiation and chemicals damage DNA, and these can cause cancer. Again, these are called mutagens because they're changed in the DNA. When the mutagens change the DNA, they're changing those instructions we're talking about. Those instructions, again, tell the cell which proteins to make. And so when you change the instructions, you can change, first you're going to change the gene, then the gene is going to make a different, potentially makes a different protein. For instance, if the instruction, we're going to use the analogy again. For instance, if the instructions were to say, if these instructions said, don't do this, when a mutation happens, that mutation is essentially saying, do this instead of don't. Or instead of saying can't, it says can. Instead of saying stop, it says go. Again, using the analogy, when the instructions are changed, they're changed in such a way that it alters the instructions and ultimately alters how the cell is going to function. With cancer, we're not concerned with all the genes because, again, there's over 20,000 of them, but we're concerned about a very specific subset of genes. And these subset of genes are called proto-oncogenes. So what are proto-oncogenes? Proto-oncogenes are latent genes, latent cancer-causing genes that have the potential under proper circumstances to convert to an oncogene. I'll explain what an oncogene here in a second. So what are these specific circumstances that we're talking about that convert or that a proto-oncogene would cause cancer? Well, we just talked about them. So mutagens. A mutagen is a specific circumstance. If these genes are exposed to a mutagen and the mutagen changes the proto-oncogene, it becomes an oncogene. Other examples would be a flaw during DNA replication or a flaw during DNA replication that is not repaired properly for DNA repair. So after exposure to a mutagen, the proto-oncogene becomes an oncogene through a mutation. So proto-oncogene means it's set up, it's ready to become a cancer-causing gene. When it becomes a cancer-causing gene, that is considered an oncogene. Each conversion from a proto-oncogene to an oncogene represents a unique mutational mechanism. Some are point mutations. We'll get into what a point mutation is. Basically, it's only when one of those bases, that like AT, is changed to something else, for instance, a GC. So only one of those that changes. And even only one change like that will potentially change the amino acid that's going to be used. When it changes the amino acid, then it changes what the protein becomes. So, and again, it's like a single word. It's like a single change from don't to do or can't to can. That's a point mutation. There's also translocation, which means, again, using our analogy, would mean getting a chapter from one part of the manual and putting it in another part and switching them out. And for instance, the one part would say, always do this. And you put it in a place that says, don't ever do this. And so you're changing the entire wording. That's an example of translocation. Translocation happens when one part of a chromosome is put onto another part of the chromosome. For instance, chromosome 9, part of chromosome 9, the arms, those looks like the butterfly, the part of the arm is lost and it's put on 22, and part of 22 is put on 9. That's an example of a translocation. So a part of the chromosome is switched out for another part of the chromosome. There's also one called gene copy amplification. So what that would mean is that instead of saying make one amino acid or only make one of something, you're making several dozens or hundreds of them, and that's gene amplification. So that's a unique conversion from a proto-oncogene to an oncogene. Again, it can be a point mutation, it could be a translocation, it could be a gene copy amplification. Mutants change or mutate genes, again, from proto-oncogenes to oncogenes. This is a very important point. As I mentioned at the beginning, all cells can become cancer. So all cells have proto-oncogenes. So that's essentially, we're all, in a sense, all our cells are able to become cancerous. again, because we all have proto-oncogenes. These proto-oncogenes are necessary for normal cell function. It's just when they're mutated or changed that they become cancer. Conversion of a proto-oncogene to a virulent oncogene represents a conversion from a normal cell to a cancerous cell. So proto-oncogenes, I think it's a very important point. I continue to emphasize this. Proto-oncogenes, when they're mutated, they become oncogenes. So what is an oncogene? We talked about a little bit about this, but let's talk about a little bit more. So an oncogene, it's a small set of genes known to be linked to cancer. Recall that genes make proteins. Oncogenes produce oncoproteins, so they're just related to cancerous cells. Oftentimes, these oncoproteins are intimately involved in regulating cell growth, as you can imagine, because one of the hallmarks, I should have mentioned this right from the beginning, one of the hallmarks of cancer is growth, excessive growth. So these oncoproteins, these proteins that are produced from oncogenes, are ultimately regulating cell growth in one manner or another. So oncogenes are formed when mutagens, these mutagens change proto-oncogenes to oncogenes, and they produce defective growth proteins, which are capable of causing cells to grow without limits. Again, this is what the hallmark of cancer is. And oftentimes, oncogenes are considered, using a different analogy, are considered the accelerators. They're considered the accelerator on a car. So they tell the car to go forward, to continue to go forward. The takeaway here is that the oncogene is the gene linked to cancer. That's onco, meaning cancer, and then the gene. So when you hear oncogene, that means it's something linked to cancer. And as I mentioned, oftentimes oncogenes are associated with accelerators. They increase cellular replication. When you increase cellular replication, you increase growth. So using the same analogy as a car, we have an accelerator. So do we have brakes? Where are the brakes? So when we go forward, there has to always, in cells, in homeostasis, there's always something that pushes it forward. There's also something that has to stop it. So where are the brakes? So those brakes with regard to cells are called tumor suppressor genes. So oncogenes accelerate cell growth. Tumor suppressor genes decelerate growth. So they inhibit growth. Tumor suppressor genes, if you have a cancer cell and you move that cancer cell, or the normal cells, you put the normal cells within the cancer cell, it'll stop this cancer cell from being cancerous anymore. And it's because the tumor suppressor genes within that normal cell are still functioning, and it'll actually normalize cancer cells. There's one example that I'll use here with regard to tumor suppressor genes is a gene called retinoblastoma gene. So retinoblastoma gene prevents cell growth. When it's mutated, when it's lost in the sense it no longer functions, then cells grow uncontrollably. And in this case, retinoblastoma is in the eye. So in this picture, on the left-hand side is a healthy eye. On the right-hand side, you have someone who's lost the function of the retinoblastoma gene, so cells are growing uncontrollably, and you see the little tumor growing on the bottom of the eye. Again, retinoblastoma is an example of a tumor suppressor gene. So oncogenes and tumor suppressor genes, that's what we've discussed so far. Oncogenes, again, turn stuff on, they make things go forward. Tumor suppressor genes block it, stop it, put on the brakes. And cancer develops not when only one of these happens, typically. So cancer develops when there's a mutation in both oncogenes, so the oncogenes get turned on, so there's accelerator. And then there's also when there's a mutation within the tumor suppressor genes, which cause the tumor suppressor genes not to function anymore. And in this sense, that's where cancer, it has to have both. It can't just have one. As I mentioned just a moment ago, is that what happens when you put cancer cells are mixed with normal cells? So for a long time, it was thought, well, cancer grows, And it's so insidious that it must be the dominant, and that normal cells must be the recessive. But what was found out again was when you put normal cells with cancer cells, the cancer cells didn't grow anymore. And so in a sense, the cancer cells are the recessive status. And that's because the tumor suppressor genes are able to normalize the cancer cells. I'll emphasize it again. Oncogenes accelerate cell growth, tumor suppressor genes decelerate. And that's why when you put healthy or normal tumor suppressor genes within a cancer cell, it basically stops them from becoming cancer. So we've talked about mutagens. We've talked about how mutagens change the DNA in both oncogenes and, proto-oncogenes, oncogenes, and then tumor suppressor genes. So we talked about mutagens, and right at the beginning when I talked mutagens, I talked about carcinogens. So carcinogens, again, carcinogens are not mutagens, but they're agents that can cause, or elements that can cause cancer. So what are examples of these? So again, carcinogens cause cancer. Examples would be alcohol, hepatitis B virus, human papillomavirus or HPV, asbestos. Asbestos causes a cancer called mesothelioma, and that's because, it affects the tissues most often in the lungs. There's also testosterone that I show here. There's also estrogen. These are all carcinogens, but again, they're not mutagens, which means they do not change the DNA. So how do they cause cancer if they don't change the DNA? That's what we've been talking about up to this point, is that it has to change the DNA. But keep in mind, these particular carcinogens, there's other ones too, promote cancer by simply causing it to have more cellular replication. So with regard to alcohol causes cells within the mouth mucosa to die. Well, you need to replace those cells. And in order to replace those cells, you have to have the current cells that are still alive replicate. And in order to replicate, they just continue to go these cell cycles. They do, I have to emphasize this again, they do not change use the analogy that we had before, they do not change the instructions. They're not mutagens. They don't change the DNA. All these cells are doing is all these agents are doing is they're causing more cellular replication. They're called promoters. Oftentimes you'll see them in the literature called promoters because they promote DNA replication. I think that another aspect to keep in mind is because if you start mixing when you start mixing carcinogens such as these, i.e. alcohol, with a mutagen, an example would be tobacco. So if we had alcohol, if someone was drinking alcohol and smoking, so the tobacco has a mutagen, so it can cause DNA changes. At the same time, you're ingesting alcohol, which causes cell death, which causes more cell turnover. When you mix those two, you typically end up with more cancer. And in the case with alcohol and tobacco, actually, you get a 30-fold increase of cancers in the throat and mouth because you're mixing carcinogens and mutagens. You're mixing carcinogens and mutagens. They say you're mixing an agent that's strictly a carcinogen, means that it's causing the cells to turn over more quickly, with a mutagen, which is changing the instructions or changing the DNA. So in a normal situation, how does a cell get the messages to grow? Because that's what we're up to this point, is that, okay, we know that mutagens cause the instructions to get messed up. We have carcinogens such as alcohol and viruses that are causing the cell to grow more quickly. Under normal conditions, what is the cell doing that causes it to grow? In normal situations, a cell is looking at a stop sign, which means stop, no longer proceed, don't grow. It's also using a pause or a yellow sign to yield, or to stop, to pause for a moment. The yellow light would be to pause, and then the green light would be to go. Now go ahead and replicate. In a cancer cell, the stop signals are lost or almost completely ignored in most cases. The pause signal is also ignored, especially during DNA replication. And for the most part, cancer cells take on a state of go, always green, all the time. So they're just moving forward. There's no pausing, there's no stopping, they're just a go, go, go. Like I said, in normal cells, the stop sign here, or these multiple stop signs, or signal lights, a normal cell is getting multiple signals all over the place saying, yes, go forward here, stop here, pause here, do this, do this. Normal cells are able to discern and make sense, okay, of these numerous signals that they're getting. Should I grow? Should I not grow? Should I pause? What should I do? That's what a normal cell does. And a cancer cell is completely different. A cancer cell is always going, going, going. Those signals that I mentioned that I just mentioned with a normal cell, those signals sometimes come externally. Oftentimes they do come externally, saying, okay, go ahead and grow because we need to replace certain cells, or we need to continue to replicate for some reason from external signals. In a cancer cell, even despite no external signals, cancer cells can continue to grow. So how do they grow more specifically? And this is where we're going to get into some molecular cell biology. This is where we're talking about what's considered a signal cascade. So how does an external signal get into the cell and tell it what to do? So we start here in a normal situation. The cell receives external signals. And in this case, it's called a ligand. And I labeled it EGF, that star. That green star is a ligand. And that's a signal. And that's a signal that's going to go to the receptor. And that receptor, again, receives information from external. So receptors basically transmit information from external to internal. External outside the cell to inside the cell. And then it's going to go through this chemical messages relay signal. So there's going to be all these little signals after the receptor, and eventually it'll end up at the nucleus, and that nucleus will do something with it. It'll make more proteins, it'll make more signals. In our case, what we're talking about cancer, basically will tell the cell to replicate. So as that ligand, EGF, binds to the receptor, that receptor is then activated, and I activate in this case. Once that receptor is activated, it causes the activation of another protein. In this case, I named it RAS. And this is a true cascade, so much simplified, but it's accurate to what it is. So that RAS is another protein. Once that protein is activated, then it'll activate another protein. In this case, is ERK. So once ERK's activated, I simplified it again, but it basically goes into the nucleus, or signals to the nucleus to do something. And that nucleus will produce transcription proteins, which are basically proteins that tell the nucleus to get ready to replicate. These are other proteins, and it'll tell the DNA to get prepped. In one sense, what it does is it causes a production of cyclin D. And cyclin D then binds to the CDK4/6. We're going to get in more detail about the CDK4/6 and the cyclin D, but keep in mind CFOS and CJUN cause the nucleus to make cyclin D. Cyclin D then binds to the CDK4/6. CDK4/6, that retinoblastoma that we talked about before with the eye and causing the tumor in the eye, the RB stands for retinoblastoma. It actually is hooked up to another protein called E2F. That E2F and retinoblastoma are in concert together. They're together. What the cyclin D4/6 does, though, is it breaks them apart. And E2F is allowed to float freely from the retinoblastoma, because keep in mind that retinoblastoma was a tumor suppressor, so it's preventing that E2F from flowing freely. When it does flow freely, it enters the nucleus and it causes proliferation. So what's the master control? If there was a master control, not one individual or not one cell or the brain isn't controlling all of this. The master control, if we were to have one, it would be the cell cycle. So through the distillation of external and internal inputs, the cell cycle modulates cellular growth. And we're going to get into the specifics of the cell cycle. Cell cycle has a G1 phase, an S, a G2, and M phase. That's growth phase 1. S stands for synthesis. Primarily, we're talking about synthesis of DNA. G2 is growth 2, and M stands for mitosis, which means the cell is going to split into 2 cells. All these 4 components of the cycle are dependent on cyclins and cyclin-dependent kinases. So that CDK that we just talked about, CDK4/6, that's a cyclin-dependent kinase. The cyclin we just talked about is cyclin D. So in order for a cell to go from G1 to S, there has to be CDKs available, and there has to be cyclins available in the right quantities and in the correct ratios in order for it to go from G1 to S, and from S to G2, and from M. So there's cyclins and cyclin-dependent kinases throughout. And we'll look at one going from G1 to S. So G1 to S. So just an overview, the cell cycle, there are 4 basic steps. The cell must move in order to divide completely. The cell starts in G1, progresses to the S phase for DNA replication, as I mentioned, then to the G2 phase, which makes sure that all the requirements for moving to M phase, and the last phase is the M phase, is mitosis, and that's where the cell splits into 2 identical cells. But for all this to happen, it has to meet numerous criteria have to be met in order for happening. And I'm going to walk you through what would be the cyclin-dependent kinase, the CDK4/6 that we just talked about. So the cyclin D binds to the cyclin-dependent kinase. I'm going to talk first, and then I'll visualize this complex, and it's a multiple protein structure. It consists of proteins cyclin D1 or D, and specifically D1, the cyclin-dependent kinase, which is 4/6, which is a serine-threonine kinase. So kinases phosphorylate proteins. So we talked about that cascade. One protein activates another one. They usually do that through phosphorylation or dephosphorylation, either one. There are many cyclin and cyclin-dependent kinase complexes that control the cell cycle and its progress. As the name suggests, CDK stands for cyclin-dependent kinases, and it is only active and able to be active, and in this case is phosphorylates other substrates, is when it's bound to another cyclin, or when it's bound to a cyclin. In the case that we're going to talk about is this case of cyclin is cyclin D. So cyclin D binds to the CDK complex. And this is essential for the progress of the cell from the G1 to the S phase of the cell cycle. And this is specifically known as the G1 to S checkpoint. This would be turning it on, and as we mentioned before, there's opportunity to turn stuff off. There's tumor suppressor proteins, and in this case we'll look at a couple. One's called P16 and the other one we'll call P53. It's called P53. And P16 in particular inhibits CDK4/6. Loss of P16 will cause cancers, notably melanoma, oropharyngeal squamous cell carcinomas, cervical cancer, and esophageal cancer. I mentioned carcinogens, viruses being carcinogens, and it's because it messes with the cell cycle. It changes the cell cycle. So let's look at this pictorially, what I just verbally discussed. So CDK4/6, again, CD is the cyclin D, and the CDK6 is cyclin-dependent kinase 6, and then you have cyclin D over on top of CDK4. Mentioned before, it basically takes off the inhibitory tumor suppressor, RB, retinoblastoma, off the E2F. That allows E2F to be free. When E2F is free, and this is what we didn't go into, which I wanted to go into a little bit more specifically here, the E2F actually goes to the nucleus and releases. E2F is a transcription factor that basically allows the genes to be expressed for cyclin EA. So just like cyclin D is for cyclin-dependent kinase 6 and 4, cyclin EA is another type of cyclin that's available, and it will bind, as you would expect, to cyclin-dependent kinase, in this case CDK2. So not CDK4 or 6, but CDK2. Well, CDK2 actually allows the G1 to pass the checkpoint into S phase. As I mentioned, there's also tumor suppressor genes. These tumor suppressor genes is P16. What does it do? It inhibits the activity of CDK6 and 4. And that prevents the retinoblastoma. It allows the retinoblastoma to still stay on the E2F. E2F then in that case cannot function. It does not release cyclin EA. Therefore CDK2 is not active, and therefore you can't go from the G1 to the S phase. You can't go to the S phase. You can't replicate DNA. You can't replicate DNA. The cell does not replicate. There's also the P53. P53 is another tumor suppressor gene. It produces P21, and P21 specifically inhibits both CDK4/6 and CDK2. In many cancers, P53 is lost. It's mutated, either through a mutagen or something happens during replication, during the replication that P53 is lost. And because P53 is lost, the cell is able to go from G1 to the S phase and become active. So in a sense, as I mentioned before, several things have to happen. You have to lose tumor suppressor genes such as P53 or P16. You also have to have activity in order for, or oncogene to be activated in order for the cell to replicate uncontrollably. So we talked about oncogenes, which turn stuff on. We talked about tumor suppressor genes, which turn stuff off. Now we're going to turn to our third category, our cornerstone of what causes cancer. We've already mentioned it a little bit. We're talking about DNA replication, and that was during the S phase. So DNA replication happens in the S phase. Cells, in order to multiply, the DNA has to be replicated, and the copy has to be exact. And it passes an exact copy on to the next cell, which is called a daughter cell. Unfortunately, that always doesn't happen. In DNA replication, there are errors. I think one thing before we get into the errors, though, to keep in mind is when DNA is replicating, it's more susceptible to mutagens. So it's more susceptible to those chemicals. It's more susceptible to radiation. So when you have cells that are replicating all the time, as in your stomach or in your bone marrow or in your lungs, that's why those are more susceptible to mutagens. That's why they're most susceptible, say, smoking. Why is cancer, not only is it in that area, tobacco's in that area, but all the carcinogens are in that area, or mutagens are in that area. The cells are already replicating quite frequently, so when they replicate frequently, the DNA is more susceptible to enter errors when it's both replicating and mutagens are present. Chemotherapy actually takes advantage of this DNA replication, so it messes with the DNA replication. Many chemotherapy, classical chemotherapy, works by attacking cells that are replicating quickly, i.e. the cancer cells, and that's how they work. And radiation, you actually use radiation therapeutically for cancer too. But let's talk about the errors. So the errors, I broke them up into three big categories. The three distinct DNA errors that can occur during replication are damage inflicted by either internal or foreign mutagens. So we're focused on foreign mutagens for the most part in the sense of chemicals and radiation, but they're also internal mutagens. The cells have a lot of energy they're producing. They're producing a lot of free radicals. Those free radicals, if the cell is trying to divide, if the DNA is trying to replicate, can actually cause mutations within the DNA. So there's internal and there's external or foreign mutagens. There's also mistakes that can be made during the replication. So there's errors. This is not a perfect system. Although it's really, really good, there still can be errors. But if there are errors, then there's a repair mechanism. But the repair mechanism sometimes doesn't works incorrectly. So that's the third. So you can either get mutagens. During replication, you can get errors, and then you can get errors if it's not repaired properly. So those are the three areas. So we've covered 4 areas now. There's the oncogenes, which can be mutated. There's the tumor suppressor genes that can be mutated. We talked about just right now, we talked about DNA replication flaws. And then we talked about DNA repair flaws. And the last couple that we're going to talk about, looks like this is a DNA, but it's in the orientation of infinity, so immortality. Cancer cells have to adjust or have to become immortal in order for them to exist. All normal cells have a lifespan, and they actually have a lifespan of the number of times that they can replicate. And that is the end of those chromosomes. There's a thing called a telomere, and that telomere continues to get shorter and shorter with the number of replications. So each cell only can replicate so many times. In cancer cells, though, they've adopted or they've taken on an enzyme. They express an enzyme called telomerase. Telomerase adds on to the end of the cancer cells. So those cells then become immortal, meaning they never die. Unlike normal cells, they only replicate so many times. All normal cells only replicate so many times, and then they stop replicating and they eventually die. So cancer cells have to become immortal, and that's how they do it, through acquiring or expressing an enzyme called telomerase. The next things cancer cells have to do, so they become immortal, but they also have to avoid a process called apoptosis. What is apoptosis? Apoptosis is basically cells committing suicide, that they have to die. Mentioned earlier about P53. Well, P53 again is a tumor suppressor protein, and it's essential because that P53 detects when there's so much damage in the cell that the cell is better off dying. So it elicits numerous other proteins, and then those proteins basically cause the cell to kill itself in a process called apoptosis. You can imagine, though, if cancer is in order to continue to grow and progress, it can't be susceptible to apoptosis, so it has to develop a mechanism to prevent apoptosis. This isn't very well defined, but it basically cancer cells do not undergo apoptosis. They get rid of all the signals, or most of the signals, that tell the cell to kill itself. So they become immortal, and then they avoid apoptosis or suicide. The take-home message for this is that in order for a cancer cell to exist, it has to go through multiple transformations. It's just not one oncogene typically. It's not the activation of an oncogene. It's not the loss of one tumor suppressor gene. It's not becoming immortal. It's not avoiding apoptosis. It's not this DNA repair going bad. It has to have a coordination of multiple things going on. Typically, there's at least 6, and I've read up to 13 genomic aberrations that have to occur in order for cancer to form. And I think the take home is that tumor formation depends on the successive and collaborative mutations. So even though you may have 13 mutations, if they're not collaborative or they're not successive, they're not in the same cascade. I only went through one cascade, but there's multiple cascades. If they're not in the same cascade, it may not even cause cancer at that point. So cancer forms after the convergence of several unlikely events. One would be, again, we've talked about these, so oncogene formation, tumor suppressor loss, DNA replication flaws, impaired DNA repair, immortality, and then last one, avoiding apoptosis. So all of these, or a combination thereof of all of these, have to occur in order for cancer to occur. So cancer forms after the convergence of several of these unlikely events. And these are the primary 6 events that have to occur in order for cancer to exist. Well, what happens after cancer exists? Then there's 2 other processes that I mentioned right at the very beginning that I'd like to talk about, and one's called angiogenesis. It's a necessary step in solid tumors for its expansion. What is angiogenesis? Angiogenesis is the blood vessels getting to the cells or getting to the cancer cell bundle in order for the cancer cell bundle to continue to grow, because cancer is using up a lot of energy, needs oxygen still, needs nutrients in order for it to grow. Without angiogenesis, cancer will remain small. Cancer bundle or group uses a lot of different mechanisms in order for it to signal out to capillaries around it that it needs more capillaries nearby. And so angiogenesis is that mechanism, or is that process by which you get more capillary growth towards the cell in order to feed it. And again, that's called angiogenesis. The other one is that once you do have angiogenesis and that cell, the capillary or blood vessel gets close enough, and the tumor can invade the capillary bed and actually puncture through it. But once it punctures through that, then the cells, certain cells, are released into the bloodstream. And this is the most devastating thing, is that once those cells get into the bloodstream, there's a process called metastasis. Metastasis, along with angiogenesis, occurs relatively late in tumor evolution, the tumor cell development. But once this step occurs, it can compromise the individual and lead to death. The vast majority of individuals do not die of their primary site of cancer. What typically is lethal is when it metastasizes, meaning when people get breast cancer, that it moves from that breast to the bone, to the lungs, to the liver. When it starts invading multiple different organs, that's what metastasis is. That breast cancer is moving to all these different sites. That's when the whole body becomes compromised. It can happen in lungs, it can happen in breast, and it's the metastasis for the most part that's the lethal component. Not very frequently is the primary site lethal. Exceptions could be in bone cancers. Those are typically lethal in and of themselves. They don't metastasize. Bone cancers don't necessarily metastasize. Only solid tumors metastasize. So the ones, as I mentioned in the beginning, lungs, GI, breast or sex organs, those will get metastasized. And they'll actually metastasize to the bone. They can metastasize to the brain. They can metastasize to the lung. And when that happens, then the whole body becomes compromised, and that potentially leads to death. So where have we gone? Over the course of almost an hour now, we've talked about where cancer comes from. If we take a longer view, it's been nearly 5 decades that cancer scientists have been looking at where does cancer come from. And we've moved from 5 decades ago, there was a lot of contention about and debate about what's the origin of cancer. And we've moved from that to being widely accepted, a detailed description of the focus and the forces behind human malignancies. And that's what I gave an overview today of. Quickly again, we'll review is that human tumors require a succession of mutations, each responsible for disturbing a distinct cellular growth control gene. The cancer cell is what is, if there is a mechanism that we can point to, it's the cell cycle. The cell cycle that serves as the master regulator of when cells grow and they don't grow, and when they stop the process. So that concludes this lecture on the origins of cancer. Thank you.